Hello, dear children. Welcome back. I'm Dr. Jisha Joe. In this session, we shall study the different stages of Prophase-I of meiosis. Now, children, before moving on to the different stages of Prophase-I of meiosis, we should have an idea of what is meiosis. Meiosis is a type of cell division taking place in the reproductive cells. Okay, that is the first thing that we should know. Where does meiosis take place? Meiosis takes place in the reproductive cells. And it is through this type of cell division that gametes are produced in sexually reproducing organisms. So, meiosis ensures the production of a haploid phase in the life cycle of sexually reproducing organisms. In sexually reproducing organisms, gametes are produced by this type of cell division, meiosis. Okay, so the first point to be remembered is that meiosis takes place in the reproductive cells of organisms, and it is this type of cell division that is responsible for the production of gametes, which are the haploid cells. Next, meiosis is the type of cell division which brings about a reduction in the number of chromosomes. Okay, so it is also called as reduction division. Meiosis is also called as reduction division because it brings about a reduction in the chromosome number of the cells, chromosome number of the organism. Okay, so we have seen that mitosis is an equational division. Mitosis is otherwise called equational division because the chromosome number of the daughter cells remain the same. Whereas in the case of meiosis, meiosis is called reduction division because the chromosome number is reduced to half in the daughter cells. If suppose the chromosome number is 4 in the normal diploid cell, after meiosis, the chromosome number is reduced to 2. If we take the example of human beings, in humans the chromosome number is 46 in the diploid cells, whereas in the gametes, that is the sperms and the eggs produced in human beings, the chromosome number becomes 23. So, it is with the help of this type of cell division, meiosis, that the chromosome number of the gametes is getting reduced. Okay, so that is with regard to meiosis. Meiosis takes place in the reproductive cells, and it is this division that is responsible for the formation of gametes, and this division, this type of cell division ensures the production of a haploid phase in the life cycle of sexually reproducing organisms. The meiosis is also called as reduction division because in this division the chromosome number of the daughter cells is being reduced to half. At the end of meiosis, 4 daughter cells are produced. In mitosis, we have seen how many daughter cells are produced at the end of mitosis. Only 2 daughter cells are produced, whereas in meiosis, 4 daughter cells are produced. 4 daughter cells will be produced. That is because meiosis is differentiated into 2. Okay, meiosis 1 and meiosis 2. Okay, and actual reduction division of the chromosomes, actual reduction division takes place in meiosis 1. Meiosis 1 is otherwise called as the actual reduction division, whereas meiosis 2 is similar to mitosis. Meiosis 2 is similar to mitosis. Okay, so at the end of meiosis 1, 2 daughter cells will be produced. These 2 daughter cells undergo meiosis 2. each daughter cell giving rise to two more daughter cells. So at the end of meiosis, a total of 4 daughter cells will be produced. I hope you have understood. That is at the end. So if suppose that is the cell at the end of meiosis 1, that is M1 is meiosis 1. At the end of meiosis 1, 2 daughter cells will be produced. Okay, and each of these 2 daughter cells will undergo meiosis 2. And each will produce 2 daughter cells. So at the end, the entire thing is meiosis. The meiosis includes meiosis 1 as well as meiosis 2. So at the end of meiosis, we get, at the end of meiosis, 4 daughter cells will be produced. 4 daughter cells will be produced. So that is in general about meiosis. Now in meiosis 1 also there are different stages like Prophase-I, Metaphase-I, Anaphase-I and Telophase-I. Similarly in meiosis 2 also the different stages are there, which are the Prophase-II, Metaphase-II, Anaphase-II and Telophase-II. So here in this session we shall study about the different stages of Prophase-I of meiosis-I. Prophase-I is a little complicated stage. It is a complex stage, and we will be studying in detail about the different stages of Prophase-I of meiosis-I. So now children, we are moving on to meiosis-I. We have already seen meiosis is differentiated into meiosis-I, which is the actual reduction division, and meiosis-II, which is similar to the mitotic division. Now we are moving on to meiosis-I. The different stages of meiosis-I I have already mentioned. They are Prophase-I, Metaphase-I, Anaphase-I and Telophase-I. These are the different stages of meiosis-I. And again, as I told you before, Prophase-I is a complicated phase, and it is the longest phase of meiosis, and it is differentiated into various stages, and we are moving on to study the different stages of Prophase-I of meiosis-I. So we shall see what are the stages of Prophase-I. Prophase-I is divided into 5 stages. Okay, first stage is Leptoton, Zygote, Pactine, Diplotine, and the 5th stage is Diakinesis. So these are the different stages of meiosis-I. Okay, Leptoton, Zygote, Pactine, Diplotine and Diakinesis. Now we shall take each stage and study in detail what is happening in each of these stages of Prophase-I of meiosis-I. So what is the first stage? The first stage of Prophase-I is Leptoton. Okay, just before the start of the meiotic division, as we have seen in mitotic division, there is an interphase stage, and this interphase stage is divided into 3 stages: the G1 phase, the S phase and the G2 phase. I hope you remember all that. So similarly, a cell which is about to undergo meiotic division is also undergoing the interphase stage. During the S phase of the interphase stage, what is actually happening? The duplication of the DNA is taking place. The DNA replication is taking place, or the DNA is making copies of itself. So again, here also a cell that is about to undergo meiosis is also performing that. in the S phase of the interphase. So there is an interphase stage in a cell which is about to undergo meiosis. Just as a cell undergo mitotic division, just as a cell which is undergoing mitotic division is having an interphase stage, here also a cell which is undergoing meiotic division will also have an interphase stage. Interphase stage is actually what? It is the growth phase. It is the stage during which the cell grows and prepares itself for the cell division stage. So here also during the growth phase or during the interphase, there is the DNA duplication taking place just before meiosis I. Okay, so remember the interphase stage is there before meiosis I. Now the cell is entering into the Prophase-I of Meiosis-I, and the first stage of Prophase-I is Leptoton stage. And I am taking a cell having 2 pairs of chromosomes. Okay, I am taking a diploid cell having 2 pairs of chromosomes. That means there will be 4 chromosomes in it. Okay, when we take 2 pairs in a cell, if there are 2 pairs of chromosomes, that is, in each pair, one of the chromosome is coming from the father and the other chromosome is coming from the mother. So there is a paternal chromosome coming from the father and the maternal chromosome coming from the mother make up one pair. Similarly, there are in such a way there are 2 pairs of chromosomes in the cell which we are going to study. So this is the Leptoton stage. So remember, when you hear the term leptoton, it is the thin thread stage. Okay, thin thread stage, because in this stage the chromosome appears as thin threads. Okay, so that is why it is called as the thin thread stage. So that is a cell. I am drawing the cell. Okay, that is a cell membrane. It has the nucleus, the nuclear membrane, and you can see the chromosomes. As I told you, we are taking 2 pairs of chromosomes. Okay, I have taken 2 pairs of chromosomes and 2 different colors have been used to represent the paternal chromosome and the maternal chromosome. Okay, so 2 different colors represent the chromosomes coming from the father, and one color, that is, suppose red color is the paternal chromosome, green is the maternal chromosome, just to represent that they are the chromosomes coming from the father and the mother. So here in Leptoton stage, the nucleus is large in size, the nucleus enlarges in size, and the chromosomes appear as thin threads inside in this stage. The chromosomes are in the process of condensation, and the nucleus is large in size. So this is the Leptoton stage. So what is to be remembered in Leptoton stage is, it is the thin thread stage. The chromosomes are in the or have the appearance of thin threads, and they are in the process of condensation, and the nucleus is large. Nucleus increases in size during this stage. Okay, and here this is a cell having 2 pairs of chromosomes. 2 pairs of chromosomes means there are 4 chromosomes, and each pair will have 1 paternal chromosome and 1 maternal chromosome. Okay, so that is the Leptoton stage. Now moving on to the next stage, second stage of Prophase-I, which is Zygote. Okay, so that is the second stage of Prophase-I, Zygote. when you hear the term zygote, remember the process taking place is pairing. So paired threads. Okay, here paired, the threads refer to the chromosomes, that is all. So here in zygote, the chromosomes appear in pairs. That is why I told you to remember the term paired threads. Here pairing of the homologous chromosomes is taking place. What are homologous chromosomes? Homologous chromosomes are chromosomes coming from the parents. That is, the peculiarity of these chromosomes is that a pair of homologous chromosomes will be similar in size and shape, and one will be coming from the father and the other from the mother. The chromosomes of similar size and shape, one coming from the father and the other from the mother, is called as a homologous chromosome. Okay, so once again, homologous chromosomes. What are homologous chromosomes? Homologous chromosomes are chromosomes of the same size and shape, one from the father and the other from the mother, are called homologous chromosomes. Okay, so here I am again drawing a cell at the zygote stage, that is the cell membrane. Okay, that is the cell membrane. Here also you can see the cell membrane, that is the nuclear membrane. Here also you can see the nuclear membrane. Okay, and you can see the homologous chromosomes getting paired. See, pairing of homologous chromosomes taking place. What is pairing? The homologous chromosomes come close together and lie parallel to each other. They lie close together. This process of lying close together of the homologous chromosomes is called as pairing. Or it is otherwise called as synapsis. Okay, synapsis. And this occurs by the formation of a proteinaceous complex between the two chromosomes of a homologous pair. A proteinaceous complex is produced which hold the two chromosomes of a homologous pair together. This proteinaceous complex is called as synaptonemal complex. Synaptonemal complex. Okay, so here in zygote, remember what is the term to be remembered in zygote? Paired threads. Remember what happens in zygote? Pairing takes place. What is pairing? The homologous chromosomes come and lie close together. This is brought about by with the help of certain proteinaceous complex, and this protein complex is called as the synaptonemal complex. Okay, and the term for pairing, biological term for pairing is synapsis. Synapsis is brought about by synaptonemal complex. Now, I am just enlarging the chromosome there. If you closely look at the chromosome, you can see that each of that chromosome, okay, each of those chromosomes that are present there, each chromosome will have two arms. Okay, so the chromosomes will have this appearance. Okay, if you just closely look at the chromosomes there inside the nucleus, you can see that each chromosome will have two arms, and the two arms are joined at the region which is the centromere. So that is how the chromosomes will look like if you look if you have a closer look at the chromosomes. This is the way the chromosomes are present inside the nucleus. Because already we have seen that before this Prophase-I of Meiosis-I, the cell has undergone the interphase stage, during which the DNA duplication has taken place. So each of the chromosome will have two arms which are called as the sister chromatids. The sister chromatids will be held at the centromere. So this is how the chromosomes will appear during the zygote stage. Each chromosome will have two arms joined at the centromere, and the homologous chromosomes will come and lie close together. This coming and lying close together of the homologous chromosomes is called as synapses or pairing, and synapses is brought about by certain proteinaceous complex called synaptonemal complex. Okay. So the in the leptoton stage as well as in the zygote stage, the chromosomes are getting the condensation process of the chromosome is going on. Okay, so remember, in the leptoton stage it is the thin thread stage. In next stage, that is the zygote stage, it is the paired thread stage where pairing of homologous chromosomes take place. This is brought about by the synaptonemal complex, and a tetrad here, as you can see, a tetrad or a bivalent is formed as a result of pairing. Okay. Now moving on to the third stage of Prophase-I, Pactine. Okay, what is Pactine? Pactine. Pactine stage is the thick thread stage. Thick thread stage. Okay, thick thread stage. Here the chromosomes are more condensed. I am just enlarging the nucleus. That is the cell membrane, nuclear membrane. Okay. Now here the chromosomes are in a more condensed state, and crossing over between the homologous chromosomes take place. Okay, crossing over see, crossing over, the chromosomes are the pairing is maximum, synapses is maximum there, chromosomes lie very close together, and there is exchange of genetic material between the non-sister chromatids of the homologous chromosomes. Okay. So here in this I am showing exchange of crossing over only at only in one pair of the homologous chromosomes. It doesn't mean that it takes place only on one side and only one crossing over can take place. No, it is not like that. Several crossing overs can take place within a chromosome. Crossing over can take place in between these two arms or between these two arms. So it is not a specific thing that crossing over can take place only on specific arms of the chromosome. Here I am just showing only one crossing over that is taking place here. Okay, now in between I have mentioned about non-sister chromatids. We have already studied in one of the previous videos what are sister chromatids. Sister chromatids are chromatids, or the arms of the same chromosome are called sister chromatids. So these two red lines which represent the two arms of the red chromosome. Suppose if that is the paternal chromosome, the red lines, the red arms are the sister chromatids of that particular chromosome. The green arms, the two green arms, they are the sister chromatids of the maternal chromosome. Okay, now what are non-sister chromatids? So one red arm and one green arm. That is one chromatid of the paternal chromosome and one chromatid of the maternal chromosome. They are the non-sister chromatids. So here you can see exchange of genetic material or crossing over is taking place between the non-sister chromatids. Non-sister chromatids of the homologous chromosomes is undergoing the process of crossing over. So remember, in Pactine stage is the thick thread stage where the chromosomes appear as thick threads when compared to the Zygote stage. You can clearly see that the chromosomes are more condensed and they appear as thick threads, and the thick thread formation is due because they are lying more close together. Pairing is maximum, or synapsis is maximum there in Pactine stage. And what has happened during Pactine? Exchange of genetic material between the non-sister chromatids of the homologous chromosomes have taken place. Okay, this is called as crossing over. So what is another thing to be remembered in Pactine is that crossing over takes place during Pactine stage. Crossing over, what is crossing over? Crossing over is simply the exchange of genetic material between the non-sister chromatids of the homologous chromosomes. So remember, it is during the Pactine stage crossing over takes place. And this is brought about with the help of an enzyme called recombinase. Recombinase enzyme is involved in the process of this exchange of genetic material between the non-sister chromatids of the homologous chromosomes. That during the Pactine stage, that is the process of recombination is taking place. Okay, genetic material is being exchanged between the chromosomes of the homologous pair. So this recombination is brought about by the enzyme recombinase. That is all what is happening during the Pactine stage. The next stage is the Diplotine stage. That is the fourth stage of Diplotine, fourth stage of Prophase-I, the Diplotine stage. Okay, two thread stage, two threads. So in the Diplotine stage, now the chromosomes in the Zygote stage have come close together, pairing has taken place, and exchange of genetic material has taken place in the Pactine stage. And now what is supposed to happen? The chromosomes of the homologous pair start to separate, except at one region, that is the region where the crossing over has taken place. That is the chiasma or chiasmata. Okay, so in Diplotine stage, the members of the homologous chromosomes move apart. Okay. See, they move apart except at the region where crossing over has taken place. This X-shaped region, the point where crossing over has taken place, that region is called as chiasma or chiasmata. Okay. So here in Diplotine, the chromosomes which remain together during the previous stage now separate or move apart except at one region, that is the chiasma. Okay, so this separation of the chromosomes of the homologous pair is due to the dissolution of the synaptonemal complex. It was this proteinaceous complex that held the chromosomes of the homologous pair together. Now crossing over is over. Now these chromosomes move apart. How do they move apart? Apart by the dissolution of the synaptonemal complex. Just draw the other pair also. Okay. So in Diplotine stage, remember the chromosomes appear as two threads. That is why, to remember easily, I am giving these terms there. Thin thread stage, here the chromosomes appear as thin threads, paired threads, because they appear as appear in a paired manner. Here thick threads because they are seen as thick threads. Then in Diplotine stage, what happens? The chromosomes of the homologous pair separate. How do they separate? By the dissolution of the synaptonemal complex. Okay, but at one region they remain intact. Which is that region? The region of chiasma or chiasmata, the region where exchange of genetic material has taken place. At that particular region, they remain together while the other regions move apart. So that is what to be remembered. What is to be remembered? The homologous chromosomes separate by the dissolution of the synaptonemal complex except at the chiasma. I hope that much is clear. Then moving on to the fifth stage, that is diakinesis. Okay, diakinesis. Now in diakinesis, terminalization of the chiasmata take place. Okay, diakinesis, terminalization of the chiasmata take place. So diakinesis, it is the last stage of Prophase-I, and the next stage is Metaphase-I. So by the end of Prophase-I, what is happening? The nuclear membrane and the nucleolus start to disappear. So that is why I am drawing it in dotted line. Okay, and the exchange of genetic material is complete. Terminalization of the chiasmata take place, and I am drawing the chromosomes here. That is one chromosome there, the other member of the pair. Okay, drawing the other member. So exchange has taken place. See, you can see. Did you see? In the red chromosome, you can see a part of the green there at the end. In the green chromosome, you can see a part at the end. You can see the red colored region. So exchange of genetic material is complete. Terminalization of the chiasmata has taken place. Exchange of genetic material is complete. Okay. And by this stage, the chromosomes are in the highly condensed stage, because the next stage is the metaphase stage. At the end of after Prophase-I, the cell is entering into the metaphase stage, Metaphase-I. So the chromosomes are, we have already seen in mitosis, the chromosomes are in the most condensed state in the metaphase stage. So here, I'm just going and drawing the other chromosome also. Here we have not shown any crossing over there, so okay. So this is the which is the stage? This is the diakinesis. Diakinesis is the last stage of prophase-I, and during this stage terminalization of the chiasmata take place. The genetic material has been completely exchanged between the non-sister chromatids of the homologous chromosomes. The chromosomes are in the most condensed stage, and since this is the end of prophase-I, the nuclear membrane and the nucleolus start to disappear, because the next stage is the metaphase stage. So that is the disappearing nuclear membrane, that is the cell membrane there. This is the disappearing nuclear membrane and the nucleolus. Okay, you can see the nuclear membrane and the nucleolus disappearing. So this is what is happening during diakinesis. Okay, so remember, in diakinesis what is happening? Terminalization of the chiasmata is taking place. Homologous chromosomes get completely separated and exchange of genetic material is complete. The chromosomes are in the most condensed stage, and the nuclear membrane and nucleolus start to disappear and the spindle fibers begin to form. Okay, so these are the things that are happening during diakinesis. So remember these terms. When you hear the term leptoton, thin thread stage, zygotine, paired threads, remember pairing or synapsis takes place during zygotine stage, and this is brought about by the synaptonymal complex, which is a proteinaceous complex formed between the two chromosomes of the pair. Then Pactine stage is the thick thread stage, where the chromosomes appear as thick threads. Pairing is complete and crossing over takes place during Pactine stage. Remember, crossing over takes place during Pactine stage. The fourth stage is the Diplotine stage, which is the two thread stage. The chromosomes get separated except at the chiasma. Chiasma is the point of crossover, the point where crossing over takes place. That point of crossover is called as the chiasma or the chiasmata. And the fifth stage of prophase-I, which is diakinesis, what is happening? The crossing over is complete, terminalization of the chiasmata take place, exchange of genetic material is complete, chromosomes are in the most condensed stage, nuclear membrane, nucleolus start to disappear, spindle fibers begin to be formed, because the next stage is metaphase-I. Okay, so we will study about metaphase-I and the remaining stages of meiosis in the next video. I hope this session is clear to you, children. If you have any doubts regarding what we have studied today, please ask your doubts and give your comments in the comment section. Thank you.